1/3 of Americans are obese. Another third are considered overweight as defined by the Center for Disease Control. The medical and economic costs of the associated health risks like heart disease and type 2 diabetes are astronomical, and in my opinion the greatest threat facing our nation. However, this trend has become more normalized in our society and continues to become more prevalent. It leads me to ask why, when it is thought that these conditions can be improved by two things: changing diet and exercise habits. It's so easy to say, but much more difficult to put into practice. While the food we eat has direct health outcomes, there are also indirect effects that differ between people, mediated by the collection of microbes that live in our digestive systems, the human gut microbiome. Diet has been shown to be the greatest driver of microbiome composition. Understanding and using this relationship can be key to preventing disease. Like many Americans, I struggle every day to maintain a healthy lifestyle in the face of so many low-cost, high-sugar food options, and I believe it's simply not practical for everyone to completely reinvent their diets, which has led me down a new research path. Instead of trying to change human behavior, what if we change the food? Since the beginning of civilization, breeding and agriculture has centered on one thing: crop improvement. And we, meaning humanity, have made enormous strides, making the plants we grow for food produce more and resist drought and disease. In the modern era, with modern problems, we must change our breeding practices so that while feeding the world, we can also feed it well. My research group proposes a new target for improving the nutrition of crops: how molecules in plants are utilized by the human gut microbiome. More and more about the relationships between the human gut microbiome, diet, and disease is uncovered every day and is extremely complex. Our goal is to illuminate the association of gut bacteria in the chemical composition of food, and further to use the way gut bacteria respond as a feature to be improved in future crops. For example, there is a fiber molecule called arabinoxylan found in the cell wall of corn that is incredibly difficult to digest by you and your microbiome. This same molecule, derived from other plants, has been shown to be a food source that stimulates the growth of gut bacteria associated with health, the good guys, so to speak. But with this molecule in corn, the positive effect is not so apparent. Because of a slightly different composition of the fiber, there are linkages between fiber chains that keep the good guys from metabolizing it. I aim to break just a few of these linkages by deactivating key genes responsible for creating them, which will lead to corn fiber that is more digestible by gut bacteria. I hope this research will change the very composition of food that everyone has access to, leading to the tortilla chips and popcorn of the future, a new generation of food for health.